Hello, my name is Andreas Grigorjew. I'm a PhD candidate at the University of Helsinki, and I'm presenting the poster of the work Sensitive Inference of Alignment-Safe Intervals from Biodiverse Protein Sequence Clusters using Emerald. Emerald is a tool that calculates so-called safety windows of proteins. We start with a protein cluster where one of the proteins is a representative and all the other proteins are called cluster members. We just define this. And then we align every pair of cluster representative and cluster member, as you can see here on the right. So looking at the right picture here, we explore the suboptimal alignment space. Sequence alignments are in one-to-one correspondence to source-to-sink paths in this alignment grid graph that you can see here, where longest source-to-sink paths corresponds to optimal alignments. The source is the top left node and the sink is the bottom right node. Vertical and horizontal edges correspond to gaps, where diagonal edges correspond to matches between two amino acids. You can, for example, see this really well with the orange alignment. This is the first suboptimal alignment on the list. And you can follow the path of the orange alignments on the left. So we are calculating what we call safety windows. You can also imagine them as conserved regions of the protein. A path in the alignment grid is called safe if it is contained in an alpha fraction of all suboptimal alignments. And safety windows are those safe paths that you cannot extend on the left and right. So for example, here we have 3 safety windows. 2 of them are present in all suboptimal alignments, where the middle one is present in 75% of all alignments. And you can see this really well on the right side, of course, but also on the left there are some paths that do not touch the middle safety window. If alpha is larger than 50%, then it turns out that all safety windows are in one alignment. And this means that we can also project the safety windows onto the sequences. Our envisioned goals is to improve the sensitivity of identifying relationships between features of proteins. We want to annotate the biodiversity of proteins on a tree of life scale. And we have a large diversity and many species, up to 100,000. And one nice goal would be to also increase our understanding of protein folding, for example. So what are the advantages of the tool Emerald? Well, it has a fast runtime, it has a good computational complexity. It analyzes the alignment space with a complexity of n squared, where n is the longer sequence length. And we also analyze the whole suboptimal alignment space rather than fixing one particular optimal alignment, because there might be several different optimal alignments, and also the scoring scheme might not be super accurate as well. And we also have a good performance in lower sequence identity ranges, as we will see in a second. So we have run some experiments on the SwissProt database, and those are all the structural amino acid types that we assigned to the amino acids in this database. And you can see here in this graph the safety coverage of each of the structure types. By safety coverage, I mean the proportion of safe amino acids with respect to all the other amino acids. And we can see here that what we called stable structure types and unstable structure types. For the stable ones, we have a higher safety coverage than for the unstable ones. And at the bottom here, we have the amounts of each structure types. So we see here that even if bridges only takes part in 0.8% of all amino acids, it's still captured quite well. Here we see the stable structure retention, which is defined as the proportion of safe and stable amino acids with respect to only the stable amino acids. As we can see here, the stable structure retention is generally larger than the safety coverage. especially looking at the lower graph here, which contains only the sequences with up to 80% safety coverage. We can really see here how much larger the safety coverage, the stable structure retention is, especially in identity ranges of 40 to 70% or 70 to 100%. A larger stable structure retention implies that the proportion of stable positions inside the safety windows is larger than the overall proportion of stable amino acids. All right, I hope you enjoyed this little talk. This is my email address here, as well as the GitHub link of the tool, Emerald, and the DOI link of the bioRxiv paper, as well as 2 QR codes to also send you to these places. I'm looking forward to meeting you at ISMB.